Hi, welcome to another lesson with Dr. Hill, and we're talking today about biology, and we're talking about genetics. So in this lesson what I want to do is describe the difference between dominant and recessive alleles. Now remember, an allele is a version of a gene. So when we talk about alleles, we're generally talking about variants of a single gene of which any individual can only have two copies. They get one from their dad and they get one from their mum. And so all of us carry two copies of every gene because we're diploid. We have two sets of chromosomes, one from dad, one from mum. Okay? So what is the business about dominant versus recessive? Well, what happens sometimes to genes and bits of DNA is mistakes are made during the copying of them, and that mistake can sometimes get into a gamete, and that mistake can be carried on into another individual. And then if it gets into the gamete, that mistake becomes part of that individual, and that individual will make gametes that include that mistake. Okay, now that sounds a little bit complicated. So let's just think about the mistake in the gene, in the DNA. If you get a mistake in a gene, what's going to happen? Well, it very possibly isn't going to make the right protein. The transcription to the RNA is going to work all right, and then maybe the RNA will go down and start making a protein at the ribosome, but the protein is going to come out differently. And if the protein comes out differently, it's going to be a different shape, and if it's a different shape, it's going to have a different function, and it maybe have no function at all. It may be dysfunctional. So if you get a mistake in a gene, you get a copy of the gene that's going through the population not doing the right thing. Sometimes it just doesn't work at all. Sometimes it's doing something badly. The shape is just varied slightly. And it doesn't do it as well as what we might call the normal, or what they call in genetics, the wild type gene. So meanwhile, in the rest of the population, we still have the gene working, and that allele being passed down through generations. Now eventually the offspring of the individual that had the mutated gene are going to meet the offspring of the individuals that have the normal gene, and they're going to fall in love and make baby mice or whatever. And then we see what happens when these two genes, these two alleles, come back together again in a single individual. And the chances are that because the normal gene, the wild type gene, is making the protein, it will make the protein, and we will see a normal tail. It doesn't matter if the other copy that they've got doesn't make the protein. It won't affect it, because as long as the protein is made by the normal gene, it will make the tail. It will do the normal thing. The protein is there, it's going to work. The only time when you're going to have a problem is if you get two copies of the one that isn't working. Because if you get two copies of the one that isn't working, you get a dysfunctional version from your dad and you get a dysfunctional version from your mum, you're not going to be able to make the protein. So whatever that protein does isn't going to happen in you. So we've been talking about tail length in mice in this discussion so far about our genetics. And so, for example, if we talk about the tail length gene in mice, that tail length gene has two possible alleles. One is a normal allele. It makes the normal protein. And when that normal protein is there, the tail will grow normally. It doesn't need a lot of... as long as it's there, it's going to make the protein, and the tail is going to grow normally. Right? So we call this big T. The reason we call it big T is because it's dominant. That allele will always take effect if it's in the individual. The mutated version of the tail length gene in mice doesn't make the protein properly. It makes a poor copy of the protein. The tail grows a wee bit and you get a stunted tail mouse. Now, that doesn't matter in an individual that's got both of these, if it's got a normal gene and a dysfunctional gene, because the normal protein will take over and just make a normal tail. You don't see it. You don't see the dysfunctional gene, the dysfunctional allele. But if you get an individual that has two dysfunctional alleles, both of the copies of its gene are dysfunctional, then it can't make the protein, and the mouse tail will not grow. So what does this mean? This means that if you're homozygous for big T, you're going to have a long tail. If you're homozygous for little t, you're going to have a short tail because you can't make the protein, it doesn't work, and your tail doesn't grow properly. If you are heterozygous, you've got big T on one set of chromosomes and little t on the other set of chromosomes. You don't get half a tail, you just get a normal length tail because the big T allele is going to make a normal protein, and that normal protein is going to make a long tail. So let's just have a look on the board here and we'll see how that is summarized. So I've just summarized everything that I've just told you on the board here. Dominant and recessive alleles, the tail length gene in mice. Big T we just call it big T. That's the allele that's functional, and it makes a normal protein and the tail grows normally. Little t is dysfunctional. It doesn't make the normal protein. The protein is a slightly different shape. The tail doesn't grow well during development. And so this has the potential to produce mice with short tails. But let's look at the genotypes. I could have a big T, that is long tails, right? I could have a big T, well, the little t doesn't do anything, it doesn't make a very good protein, but the big T makes a normal protein, so we're going to get long tails. And the other possibility is little t. And in this case, I don't have the normal gene in my genetics. I don't have it in my genotype. Therefore, I can't make the protein. Therefore, I only make the dodgy protein, the dysfunctional one, and I get a short, stumpy tail. All right. Now, who knows? A short tail might be an advantage someday, and it may be that these become the dominant type eventually, because, I don't know, if some hunter comes along and it attacks tails, a short tail is better than a long tail. So there's no better or worse situation. It's the environment that decides which genotype for any particular gene is better or worse. But in the meantime, what do we get? So our big T is dominant, and this is what we call our phenotype. And this is what we call our genotype. We've talked about genotype before, but phenotype is what the organism actually looks like after the cross has been done. The effect of the genotype. So the effect of a big T is long tails. The effect of big T is long tails. The effect of little t is a short tail. So these two genotypes both produce similar looking mice. It's only this genotype that produces something different. Remember, the dominant allele will always happen. It will produce its phenotype if it's there. It's only when it's not there that you produce the other phenotype. So, I hope that was clear to you. If it wasn't clear to you, have a look at the video again, watch the video, listen to the argument, and I'm going to call a halt there, because what we're going to do next is we're going to look at some more crosses, and we're going to calculate the probability of different genotypes coming out of the cross and the probability of different phenotypes coming out of the cross, because that completes our learning for looking at one cross. All right, so I'll see you in the next lesson.